Thanks so much for having me today. I'm so excited. I have to be honest, it's amazing to have the opportunity to speak with you guys this morning, to tell you a little bit about the work that we're doing at Moravian in my research lab. Now, a lot of what Justin said, depending on if you studied science while you were here at Moravian or you didn't, it might have sounded like a lot of mumbo jumbo, but it is return to learn today. And so the good news is that my job is to try to break it down for you so that whether or not you studied science at Moravian, you are going to now be able to engage a little bit in what we're doing in our lab. And so I was wondering if I could get a quick maybe response from the people who are attending. At the bottom of your screen, you should see a reactions button. And so if you click on reactions, you'll be able to give a thumbs up or a round of applause. If you studied, let's say biology or chemistry or yeah, Robert, okay, I feel you, Robert. I know that Robert was here just with me a couple of years ago. If you studied biology or chemistry or neuroscience when you were here, could you go ahead and do the round of applause function real quick through your reactions? Nice, Amanda, great. Well, Pearl, I was actually gonna say if you didn't end up studying science when you were here at Moravian, if you give me a thumbs up real quick so I can see those non-scientists who are here ready to sort of open their minds and learn something new, which is so exciting. This makes me even more pumped. Yeah, so that's wonderful. We've got a great mix. And I think something today, in order to sort of set the stage for you, is that we're in a global pandemic, right? And I think in terms of how it's changed our lives, it's in an amazing way. Not amazing good, but just like we never would have thought that it would be like this. And so when I start to talk about, our research kind of relating chemistry to bacteria and infectious disease, it's important for me to sort of pause and recognize that moment and also say that COVID is not a bacteria, right? So it's a virus, and the way that we look at them are really different. And so the conversation today, I would like to stress to you is it's of a different importance, but I would argue could be of an equal importance in terms of how it could affect our world. And so, as you all engage in this presentation today, I encourage you to ask questions that may pop up from what you may have heard about bacterial infections. To get started today, and as I like to do in my class, I usually like to get a little bit of audience participation. So I've prepared for this via Zoom. One way that we can do this is actually by using the tools that Zoom offers in terms of our participants window. You guys can actually give feedback. And so I'm gonna ask a few questions to see first what you know about bacterial infections. So some of you guys who may have joined didn't really expect to be put on the spot today. But the best way we can learn is to engage. And so hopefully you can share your answers here and we can get a little bit of background information on what you know already about bacterial infections. So I bet it's a little bit, even if it's not, your expertise or field of study. So if you look at the bottom of your participants window, you'll see that there's a couple of buttons. You've got a green one for yes, a red one for no, a gray one for go slower, and a blue one for go faster, which I guess are feedback that you can usually give to the presenter, which, I guess if it's really that bad, you could tell me to go faster or slower today, but for now, we're actually gonna use them as like multiple choice answers, kind of like A, B, C, and D as we answer these questions. So if you could go ahead and respond based on what you think is the correct answer, then we'll sort of lay the land for what we know already about bacterial infections. So here's your first question. So what type of medications do doctors prescribe to treat bacterial infections? Oops. All right, we see some responses here. It looks like the majority of us got it right. That's correct. So it is antibiotics. Very good. So vaccines, in fact, are used for treating viruses. Antibiotics are for bacteria. Chemotherapy is for cancerous eukaryotic cells. And then steroids, actually, we all produce steroids in our body, things like testosterone. But, there's also negative connotations to steroids when we think about like bodybuilding and stuff like that. But they are natural, not that we should use them incorrectly. They also won't necessarily help with bacterial infections. Okay. So let's take another try at a follow-up question. Does anybody know what term describes the evolutionary mechanism bacterial species use to survive after being treated repeatedly with the same antibiotic? All right, so let's see. The correct answer for this one is antibiotic resistance. Okay, so that one, this one's tough, but I see antibiotic resistance in this case. Some of the other terms like survival of the fittest you may have heard from an evolution class you would taken, but antibiotic resistance is specific for bacteria, and it's the ability of bacteria to actually develop biologically mechanisms that basically make antibiotics less effective. And this is pretty common. In fact, that's the final question. What percentage of infectious bacteria have developed a resistance to at least one type of antibiotic treatment? Okay, you can go ahead and share your answers there. Oops. Now, this might be surprising, and I don't want you to be scared by this response, but it turns out that 70% of bacteria are already resistant to at least one antibiotic treatment. So what that means is effectively 70% of the bacteria have already found a way to beat the antibiotics that we treat them with. That could freak you out. Well, here's the thing. We have a lot of different antibiotics, and so some of them are effective against different types of bacteria, which is important. What I'd like to do for you today is kind of show you a little bit about how this is still a consistent concern among scientists who are studying infectious disease. So I have for you a graph that shows these colored arrows. The way that we can interpret this is that the beginning of the arrows here actually talk about the start or the development of this particular antibiotic. Some of them you might recognize, like penicillin you might have heard of, okay? So this was developed, you know, just before the 1930s and the 1920s. By mid-1940s, we already had bacteria who were resistant, who could not be treated by penicillin. That's what these arrows mean. And so you can see that the development of these antibiotics, they have a lifetime. And after so many years, they become less effective. And so this is a little bit scary because a lot of these antibiotics are critical to maintain our health. And so when we see these arrows sort of end and we look towards the future, the only way that we can deal with this is to try to develop new antibiotics, because the bacterial resistances stop these antibiotics from being as effective. And so we need to come up with new ways to treat them. And understanding and controlling these pathways of infections and resistance are so important for us to actually be able to minimize the threat that comes from infectious bacteria. This is the task of our lab. This is what we're trying to do through the research here at Moravian in my laboratory. And right now, I want to actually focus on one specific mechanism that bacteria use that helps them build resistances, okay? I think when we think about how bacteria develop these resistances, it's probably really important that we first understand what causes a bacterial infection in the first place, okay? Now, of course, since we're living in the COVID world, we all know that the way that we are infected is generally first by transmission. So we have to have the bacteria somehow get on us or in us, right? So we're exposed to the bacteria first. Then they incubate within our bodies and they colonize, so they reproduce and they grow. And then actually what happens once they get to a certain amount of bacteria inside you or inside any host, they start to produce something called virulence factors. And now this, especially if you're from a non-science background, may be a brand new term. Virulence factors are basically biological molecules, proteins, if you're familiar with that term, that actually allow the bacteria to alter either its own physiology or its own makeup, or it can actually alter the makeup of the host. So in this case, you, the human that's been infected. And the types of virulence factors made, they vary. So here's just a few of them. One thing that virulence factors can do is that they can be toxic. So bacteria can actually produce toxins that then affect their hosts in negative ways and impede their sort of normal biological function. So that's dangerous. Another thing that can happen is bacteria can actually create capsules. And these capsules are protective barriers around the bacteria that make them sort of, I guess, stronger, like a shield. So they're more resistant to being, either killed or harmed. Another type of virulence factor that's produced is an advantage for the bacteria in terms of adhering more strongly to surfaces within the host. So you can think about it just like super glue, attaching the bacteria to where they're living. We also have these things that can occur called biofilms. And this is when a lot of the different bacteria within the same species, they secrete this sort of sticky substance, yes, like your neighborhood Spider-Man's web, that actually keeps all the bacteria sort of together in a clump and makes them resistant to environmental change. These are things like big changes in temperature, big changes in acidity, and it just won't harm the cells as much as it normally would if they didn't have this thing called a biofilm. Finally, another type of virulence factor increases the bacteria's ability to invade its host cells or increases its motility, so its ability to move. So again, this is all kind of crazy that bacteria are able to control all of these things. And all of these things, depending on the species of bacteria you're looking at, can contribute to the ability of the bacteria to infect a human host. Here's what's so amazing. The diversity of these virulence factors, they're oftentimes controlled by a similar process. And that similar process in this case is going to be, again, the core focus of our study. This similar process really allows the bacteria to know when to produce these virulence factors. It's important for you to consider that the bacteria cell is just one of them alone. We're humans, we're huge, right? We're made up of tons of cells. One bacteria cell is microscopic. When you get that one bacteria cell in you, it's not doing squat. No change, no harmful thing happening to you. But the thing is, when there's enough bacteria present, if they can communicate to each other and figure out when to release these virulence factors, if they can do it in a synchronized fashion, then you're in trouble. And so this process that allows bacteria to communicate and regulate this virulence factor production is called bacterial quorum sensing. So this is a process of chemical communication where the bacteria use chemical signals, which is why as a chemist I'm interested in this. They use chemical signals to communicate. So when we think about quorum sensing, this phenomenon is sometimes a little bit complicated. And so I've come up with an analogy that helps us describe how this happens in sort of everyday words. And so because this is the alumni return to learn, I thought it'd be really fun to ask for a volunteer to engage in this analogy, which quorum sensing is quite similar to the way in which we maybe plan group outings. So I'm wondering if someone could raise their hand. They could even be so bold as to turn on their video. I know that sometimes that's an uncomfortable thing to do, but I would love to engage you in this analogy. And also feel free to unmute your mic for this one. This one, maybe we can just do that and then we'll remute later on. Oh yeah, all right, good thing you're here, Lauren. Actually, this is gonna be, so Lauren's my sister, which is awesome. She's also a Moravian alum. So happy that she's here. Pearl, also, hi Pearl, how are you?
Okay.
Well,
okay.
I'll take it.
So maybe Lauren and Pearl can do this together. So I would say first, Lauren, Pearl, when you were at Moravian, right, you had long weeks of classes, you had homework. At the end of the week, you needed to do something fun, right? So if it was Friday after a long week of classes, what would you do, Pearl? What would you be doing to relax or to have fun at the end of a week of classes at Moravian?
Well, since I'm Jewish, I would not be doing anything Friday night or Saturday mostly, except going to synagogue. But Saturday night and Sunday, I would either get together with friends or watch football games or go to one of the activities at Moravian.
Oh,
But it was
awesome.
a long time ago that I was at Moravian, a very long time ago.
Well, let me tell you something, Pearl. That's even more exciting that you made it on the Zoom call and you're having this discussion with us today. So thanks so much. But I'm going to go ahead and grab on the gathering people for a football game. And so it can be even more fun to imagine a world where Lauren, my sister, is at Moravian with you. Like we just time warped and you guys are together. Okay?
It's funny.
And
Okay.
so Pearl and Lauren now are trying to coordinate this outing. Okay. So maybe we'll say that it's happening nowadays, right? Just to make things a little bit simpler. So let me turn it over to Lauren. Lauren, if you wanted to get everybody together to watch this game with Pearl, how would you communicate to all your friends at Moravian and all Pearl's friends? Say, hey, we're going to watch this football game. Let's get together at this location, you know, and watch it. What would you do?
I would probably text them.
You would text them, right? That makes sense. I think we can all agree that that's probably what we would do nowadays. Okay, great. Laura and Pearl set this up. They're planning this outing to go and see the football game. They're going to watch this football game. So Lauren picks a time. She goes, okay, I know Pearl's free on Sunday. Usually when football happens, let's all get together around noon. We're going to go down to Roosevelt's. Well, that's not Roosevelt's anymore. What is it? 21 Craft House, I think. We're going to go watch this game. Okay, let's all get together and do that. So Lauren and Pearl are waiting, you know, just at the corner by Colonial Hall, and it's about 11:50, right? They sent their messages. We assume their friends received those text messages. They also have phones, right? They got those messages. And it's like 11:50, and one or two friends start to show up. Lauren and Pearl are looking around. They're like, we sent out our message to a couple more people. Let's wait a little bit longer. Gets to 12, okay, couple more people gather. Good. So we've got now 4 or 5, but Lauren and Pearl are like, I was expecting a few more. Of course, naturally, as this happens with, you know, Robert and Diane and Marilyn often, is that we had to wait till 12:05 for them. Okay, but they finally came. And now Lauren and Pearl look around, and we got the whole group here. And then all of a sudden, what happens? Off we go, down the street, down to 21 Craft House, time to watch the game, right? This exact scenario is what the bacteria do. This is exactly what they do. Let's assume that they all have these cell phones, okay? And let's assume that they can send these text messages. Here's Lauren and Pearl. Lauren and Pearl are these green ovals, okay? They've sent their text message out. We'll say the text message is this yellow triangle, okay? They've sent their message out to their friends, just hoping they'd respond. Good news, they have good friends. They did. And so the message was sent back. People started to gather around Colonial Hall. We got a couple people there by 11:50. Before you know it, it's 12:05. Everyone's around, and Lauren and Pearl are like, all right, we got everyone. Off we go, okay? So what the bacteria are doing are they're secreting these chemical signals. They're sending out these chemical signals like text messages to say, hey guys, where are you? We got to get together. The thing that's really neat is that the bacteria that are of the same species, they have an ability to receive those messages. They could be the cell phones in this example, but really what they are they're biological constructs that recognize the signal. And then as a result, we get this event where at the same time, everyone decides, all right, it's time to go. For the bacteria, at the same time, they say, hey, we're going to infect this host now. We're going to make these virulence factors. We're going to make these toxins. We have enough of us around to make a difference. And so this is bacterial quorum sensing. And this is the process that we're actually studying. Now, here's the thing. The reason that this is so impactful is because the communication that's occurring actually leads to some of the negative effects we see in bacterial infections, these virulence factors. So imagine that I take Pearl's phone, okay, right before she's about to send that text message, and I throw it on the concrete. Phone smashes to bits. Sorry, Pearl. Didn't mean to do that to you. But guess what? Can you now send that same text message? You've lost your device, right? So now you've got an issue. What we're trying to do from a chemical sense is we're trying to destroy cell phones. We're trying to basically stop these signals, okay, from being sent amongst the bacteria. Now, the reason why this is helpful is because if we inhibit quorum sensing, we stop those virulence factors from being produced. The other really interesting thing about this too is that a lot of antibiotics kill the bacteria cells. When you kill the bacteria cells, evolutionarily, that other wrong answer, survival of the fittest, the bacteria are going to try to find a way to survive. When you're just simply stopping their communication mechanisms, they'll also still try to find a way to do that, but they're going to be a lot slower. The need for them to sort of evolve, to develop resistance to the way that they're being treated, it's slower. So by looking at quorum sensing as an avenue to inhibit this chemical communication that's linked to bacterial infections, we can start to inhibit these negative effects while also not incurring bacterial antibiotic resistance as quickly. And so that's why it's an interesting target. I'll pause in case there's any questions. I don't know if we'll save them all to the end, but this is kind of an interesting turning point in the presentation. So just to make sure everybody's on the same page. I don't know, Justin, if you feel like that's okay, but.
Absolutely, Mike. Yes, if anybody has any questions right now, feel free to put them in the chat box, and that way Mike can answer them before progressing on.
All righty.
No one? All right.
Yeah, feel free to drop them in. Thanks, Justin. I appreciate that. We'll keep going. And even if some of them come in, if they're urgent, we'll take them right away maybe. And if they don't, then we'll just save them for later on. But the thing that's really important from a chemical perspective is now we're curious what the signal is, what this yellow triangle is, okay? And if we understand the structure of this chemical signal, this text message that's being sent to the bacteria, then we can actually start to figure out how to interfere with it. So right now, it's important for me to give you a little bit of context on just how quorum sensing is used for different bacteria. So maybe the most important thing I'll show you here is that there's two types of bacteria called gram-positive and gram-negative. Now, where that name originates from is not too important, but I listed some examples of gram-positive and gram-negative bacteria here at the bottom of the screen. And so when you're looking at these bacteria, some of them you might recognize. You may have heard of bacterial pneumonia. You may have heard of Listeria, E. coli, Salmonella. Okay, so some of these bacteria are pretty common. We've heard them before. They all quorum sense. They all do this. Now the thing is that depending on the class they're in, whether it's gram-positive or gram-negative, the signaling molecule, the chemical that serves as the text message, it changes. So in gram-positive bacteria, we use very short proteins known as peptides. So peptides are made of things called amino acids and that are linked together to make sort of this longer chain to form the biomolecule. So peptides are usually made, they're about like 5 to 40 amino acids. They're a lot shorter than big proteins. In gram-negative bacteria, we don't use amino acids, peptides, and proteins. Instead, we use small organic molecules. The bacteria basically have, and I know that the image here may not really show you too much in terms of the atoms. If you've taken organic chemistry, you might recognize some of the drawing here. But basically, the organic molecule, it doesn't have any metals in it. It's usually all carbons, oxygens, nitrogens, hydrogens. And that's sort of one way we characterize the molecule in gram-negative bacteria. Our research at Moravian focuses on peptides. So it focuses on those short proteins that are used as signals. And what I'm going to do now is take you through a little bit of sort of how we've come up with our hypothesis for addressing quorum sensing chemically. Then I'll actually take you through a couple of our projects just briefly, because it gets a little bit intense in terms of the numbers and stuff. And really for this Return to Learn, our goal is just to sort of introduce you to quorum sensing as a new way to develop antibiotics. So here's our major research question. If we're looking at a way to inhibit bacterial communication and quorum sensing using chemistry, we have to know a little bit about the way that the quorum sensing system works within the cell. Okay, so I'll just highlight a couple things here. There's two major areas that we could think about attacking chemically so that we could try to intervene with either the production through the synthase of this quorum sensing molecule or we can actually inhibit the way in which the quorum sensing molecule or peptide is recognized by the bacterial cell. So we can stop the production, meaning we can inhibit at something called the synthase, or we can stop the recognition of the signal. So again, it's like destroying the phone here. We can stop the recognition of the text message. What we're focusing on in our lab is studying how the quorum sensing peptide, the molecule, binds to the cell at the point called the receptor that recognizes the peptide and basically leads to all the internal biochemistry that makes the production of virulence factors. So if we can do this and understand chemically how the peptide actually binds to the receptor, we can start to intervene here. And so that's really actually the goal of all the projects that are going on in my lab. We're looking at different strains and species of bacteria, but we're really trying to interfere with the way in which the bacteria cells recognize the peptide themselves. So I've basically taken, you can see the similar colors here. That wasn't totally intentional. Okay, so orange and red. You can see the way in which the peptide here, still shown in orange, might engage with the receptor. That's as part of the cell. And if the peptide comes together and binds with the receptor, that's when we see this increase in these virulence factors, these things that cause infection. Here's something that might surprise you. We all are amazed and in wonder about the human body for many reasons. I mean, sometimes some of us can think about we don't take care of our human body so well. Somehow we still wake up every morning and survive, right? So the thing is that the human body is amazing and it can do a lot of really cool things, but it's not perfect. It's not perfect. And when we think about the ways in which we try to have these bacterial cells in some ways responding to the binding of the peptide, the bacteria aren't perfect either. There's ways in which that we can sort of take advantage of deficiencies in the bacteria by using chemistry. So let's think about how this peptide maybe is recognized by the receptor to start quorum sensing. Oftentimes when we think about amino acids, and for anybody who did take sciences at Moravian, you'll know that generally there's 20 amino acids that are used over and over again to make up different peptides and proteins in the body. 20 is actually a pretty limiting number. And so sometimes when we think about all the different processes our body regulates, well, with 20, only choosing from 20, it's not always perfect that we have a great fit between our receptor and our peptide in this case. So what happens sometimes is that we see these peptides and receptors come together and there's, maybe spaces that don't like fill totally. And maybe there's some spaces where there's a little bit too much from the peptide to get into the receptor. So there's not a perfect match. The thing is, because we have some tools through chemistry to modify the atoms and the structures of the peptides, we can use non-natural amino acids. So literally amino acids that we make in the lab. Your body can't make them, we make them. And we can actually modify the surface of the peptide. So now this crazy shape that doesn't exist in the body, well, if we've understood the receptor well enough, we can make an amino acid variation that fits even better. This is our goal. Our goal is actually to modify the peptide to make the binding even better, because then what we can do is we can outcompete the natural peptide that the bacterial produce. If we can outcompete, then we can control things, right? We can control the ways the bacterial communicate. And this is really what we're trying to do. So again, if you have any questions about that, feel free to throw them in, or so Justin can read them off. This is another big sort of key concept to understanding really how our research is able to interfere with bacterial communication. I think the part that's coming next, I'm really excited about, because I get to show you a little bit about how we make these non-natural peptides, like literally the work that my students do. And then I'll also show you some of the different bacterial species that we've been investigating. All right, off we go. So one of the bacterial species that we're looking at is strep pneumonia. So you may have heard of bacterial strep. My goodness, you may have had bacterial strep, okay? And I think in terms of the effect that this has on sort of global health, it's kind of a big deal. Strep pneumonia is actually something called an opportunistic human pathogen. It's actually living in us right now, but it doesn't infect you unless you become immunocompromised. So you get sick, it sees an opportunity, and then it makes the situation worse, okay? So for people who have been infected with this, some of the things that result are pneumonia, as you know it, sepsis and meningitis, okay? And the source of the number of infections that we see and the source of deaths is huge. We're talking like 22,000 per year. And that costs a lot of money to deal with those infections for treatments and rehabilitation. So this is an interesting target. This is something that we need to sort of be able to work against to make sure people stay healthy. And so when we think about this bacteria and quorum sensing in this bacteria, there is a peptide that mediates the quorum sensing. This peptide, I showed you this cute little ribbon here, okay? If we were to model this, this is actually what it looks like, okay? It's about 17 amino acids long. It's a little spiral we call an alpha helix. And the name for this peptide is CSP1, okay? So if you see that name again, it's just talking about this peptide, this text message, quote unquote, that actually leads the communication in this bacteria. Now, the virulence factor that this regulates, remember the thing that causes the infection or makes the situation kind of bad for the human host, it's called competence. Competence is a virulence factor that effectively we call an invasion factor. It basically allows the bacteria to take up extracellular DNA from the environment. Now, this is kind of a weird thing, but what this does is if some neighboring strep pneumonia cells have died, whether they've been treated with antibiotic or there's been some stress event that's made them die, if these cells are competent, they can pick up the leftover DNA, kind of like clues, and figure out what went wrong. That allows them to then build new mechanisms to resist that threat to their survival. So competence is a very serious thing, and it's one of the major ways in which bacteria can build resistances to antibiotics. So we want to try to shut this down. So we got our target now, right? And so this is what we want to affect change in. We got to start to modify this peptide. Now, when we make new peptides, we call it peptide synthesis. So at this point, we're ready to synthesize. If I take you into my lab, I'm going to show you some pictures. If a student was coming in tomorrow and wanted to make a new peptide, this is what they would do. And so this is really fun to show you. We've got a microwave peptide synthesizer. This instrument allows us to put amino acids together to make a peptide pretty quickly. We can actually add a new amino acid or a new unit to a longer chain in about 10 to 15 minutes, okay? The students have named this microwave synthesizer Magic Mike, okay? And what it does, it's got a little vessel in it that we can conduct something called solid phase peptide synthesis. A lot of reactions in chemistry, and for those of you who have taken a chemistry class, you usually take some solids and liquids and you put it in a solution to dissolve it so that things react together. It's really amazing what this technique allows us to do is actually attach peptides and an amino acid to a polymer, a solid support. That solid support looks like this. You can see the little yellow at the bottom. What we can do using this technique and this technology is we can add amino acids to the polymer and then just kind of wash out any impurities or excess reagents, which means that we can add amino acids on, do a wash, add another one on, do a wash, and it's all really fast. Usually when you run a reaction, you have to do a series of purification steps that takes hours and hours. This takes minutes, and so it allows us to move pretty fast. Once you end up adding your amino acids and making that peptide on the solid support, the next thing we do is we put our peptide on Shakira. You can see the name right there. My students said name our orbital shaker Shakira with strong acid. And then it cleaves the peptide from the polymer. And so now what we finish with is this like white powder. And this is what we call our crude peptide. Not because the peptide's mean or rude, just because it's not fully purified at this point, okay? So these steps allow us to get literally any peptide we want. We can make any mixture of amino acids. We can even take those things I referred to as non-natural amino acids before, the things that we chemically modify, we can just insert them wherever we want in the peptide sequence. And so that allows us to redesign the quorum sensing peptides. After you do that, we have to purify it. I know I talked about how nice it was to wash the polymers and get them a little bit cleaner. Okay, we can do that super fast. Inevitably though, there's still going to be some products that we made through our reaction that we don't want. And so HPLC allows us to separate out the products that we don't want. So when we put our white powder, that crude peptide, in our HPLC, we might get a trace that looks like this. We've got a lot of different peaks here. All of these little peaks represent different products that were made in the reaction. So my students will inject into this instrument some of that crude peptide mixture, and they will select the peaks that hold just their peptide. And usually this takes a little bit of time, but eventually we usually see one peak, like maybe this big blue one here, that is our peptide that we desire to make. And we simply then just collect through using a detector, only that peptide. And that gives us essentially our pure single product. We then take that down to something, and this name is ridiculously long, but matrix assisted laser desorption, ionization, time of flight, mass spectrometry. MALDI-TOF for short. This instrument we have in the department, we're really lucky to have it. What it does for us is essentially identify the peptide products. The easiest way I can describe this to you is like barcodes at the supermarket. When you take a bottle of orange juice versus a gallon of milk, the checkout recognizes which one you have using the barcode. When it comes time for MALDI time of flight, every peptide is made of a bunch of different atoms. And depending on the units or the amino acids we put into it, it has a certain mass. Those of you who have taken chemistry before, molecular mass, grams per mole, this is what we're looking at. So it's very hard to see on your screen, but there's a number up here at the top. It's 2243.284. 7 numbers, think about that as being an identifier. When we see the number that we have expected for that peptide, we know we've got it, okay? And so this instrument allows us to essentially say, hey, you know what you were trying to make? Here's confirmation that you made it. So the students will go through these steps to make these peptides. They will synthesize them, purify them, and then identify them. Once they identify them, it's time to jump into the bacteria. Not literally, it's not good. I mean, it's a pathogenic bacteria, but we actually do some bacterial experiments. So for strep pneumonia, what we use is a genetically modified bacteria. Now there's some language at the top about the genetic modification, but the most important thing you should know about this genetic modification is that it allows us to track how high quorum sensing is turned on by a change in color, okay? So here's how we can think about it. If we introduce our peptide, the quorum sensing signal that we made, and it doesn't turn on quorum sensing very well, then we'll see very light yellow coloring. If we have a really good fit between our peptide and its receptor, what will happen is that the bacteria will emit a yellow color, and we'll see a dark yellow orange in our assay. And that'll let us know we've got a peptide that really fits well. We can then take these color differences and we can analyze them using an instrument. We call it a plate reader for short, but essentially what it does is it kind of reads the differences in colors. From the differences in colors, we can get numbers called absorbance readings. And from these absorbance readings, we can actually get values that tell us how potent or how efficient our peptide is at activating quorum sensing in strep pneumonia. So I'm going to show you these numbers in a second, and there's going to be a lot of numbers on here as I take you through this. This is sort of when we get a little bit more technical, but just keep in mind here that when you're looking for the numbers called EC50s, the lower the number, the better. That means we're activating quorum sensing more. So here's the peptide sequence. You can see at the very top, if you've done biochemistry, if you've done biology, you might recognize a single letter amino acid code. If you haven't done any of those science classes, all you want to notice at the top is that every letter that you see represents a different amino acid or a different construct in the whole peptide itself. We've realized through our previous research that a couple of these amino acids, L, I, and L, shown in red, are really important for binding to the receptor. So we started by doing our experiments there. The I's and the L's actually represent two amino acids called isoleucine and leucine. Again, if you didn't have chemistry or biology before, I just want you to notice that there's small differences in the shapes at this part of the molecule, okay? What we did is we actually made some substitutions. We took amino acids that you can't get, okay, biologically. These, especially at the bottom, you'll see the changes in structure, the shapes are different. And when we change the shape of something, just like if you remember from that image where we showed the peptide coming into the receptor, when you change the shape, okay, it's recognized differently. And that's what we were trying to do. And so these non-natural amino acids, we actually put into some of these positions shown in red. And that was allowing us to see a difference in quorum sensing ability. So there's a lot of numbers here, but I've got a way in which I'm going to show you just a couple that are really important. So you'll see the bar that's appeared in red. If you notice, there's something called an EC50 value for the one in red, and it says 10.3. 10.3 represents how active quorum sensing is in strep pneumonia with the peptide that the bacteria makes itself, CSP1. So that's like our baseline. That's what we're comparing to. If we could find something that gives us an EC50 value that's less than 10.3, that means we're activating quorum sensing better than nature's doing it. We're activating quorum sensing with our manufactured peptides better than the bacteria itself. It turns out that we have a few of these substitutions that allow us to do that. You'll see numbers at 5.74, 6.68. That means the peptides that my students have made in the lab are actually activating quorum sensing twice as good as the bacteria themselves. So this is significant because this means that we are actually able to bind to the receptor better than the natural peptide, because these values that we're reading through our absorbance assay, which was making the bacteria yellow, we're seeing a difference. Isn't that great? We can actually increase how this bacteria communicate. We can make them communicate better, right? Mm-hmm. Right now you should be like, no, don't do that, okay? These bacteria are bad. What are you trying to do, okay? Like, I thought we were trying to prevent this from happening. Well, you're right, we are. And we're not some diabolical villain, okay, though we're close to Halloween, and I've thought about it. No, I'm just kidding. We're not some diabolical person that's trying to actually increase chemical signaling of a bacterial pathogen. We're actually getting this information so we can do just the opposite. Here's the kicker. There's one of these amino acids, and it's the letter E up here. Stands for glutamic acid. If you don't have a biochemistry background, do not worry. All you need to know is that this one amino acid, if we switch it out, if we change its structure, and we take away this group here at the bottom, it changes the activator into an inhibitor. So it stops the quorum sensing as opposed to increasing the quorum sensing. So we already had this information. So really, if I can explain to you what we were trying to do, we were trying to find a way to bind the receptor better. We put those non-natural amino acids in to make a more tight, stronger attraction to the receptor. Knowing that once we got the stronger thing and outcompeted the natural peptide, we were going to make a little switch at the side that was going to take it from activating and make it an inhibitor. And that's exactly what we did. And so here, the IC50 value, which is different from the other one before, this shows how well we're inhibiting quorum sensing. And with this one change here, represented by E1A, you're going to see numbers again where the lower number means better. The lower number, we're doing more inhibiting of quorum sensing. And we now have made a series of quorum sensing inhibitors for strep pneumonia. Even one that gets down to 54, which was, at the time this was published, the most effective quorum sensing inhibitor in the field, like in all of the quorum sensing research for this particular bacteria. So we're really excited about that. So with this information, we're continuing our work on strep pneumonia, but it's not the only bacteria that we're working on. So I'm going to finish the presentation today, probably about 10 more minutes, just showing you a couple of the other projects that we're working on. But again, I'll pause in case there's any questions for people who are listening. Feel free to just drop them in the chat. And also, you're going to have to respond to some other questions coming up soon. So just like in class, if you were zoning out, you might want to come back to me real quick so you can answer those questions in a few minutes. All righty, here we go. So in addition to strep pneumonia, oh, we got a question. Robert. As an aside, many of the ultimate deaths from COVID-19 were for Robert, you're absolutely right. What Robert points out here is something called a comorbidity. So what he's realizing is that because COVID-19 has reduced basically our ability once we're sick to fight bacterial infections, that just like we talked about before with strep pneumonia as an opportunistic pathogen, once people are sick with COVID-19, it may be the bacterial infection that kills them, not the viral infection. That is true. There's more research coming out, and it's very recent, that's found this to be true. Not all the time, but there's a possibility that that's the real cause of death for someone who's sick with COVID. Nice. Not nice. Definitely not nice, but very insightful point. Great. Okay. So let's continue on then. The two other species we're working with, one of them is also a pathogen, but it's a different kind of pathogen. Here's a picture of it. Yeah. You guys may know that, yeah, bacteria can cause something like pneumonia, okay, which leads to a respiratory infection. And maybe you don't want to raise your hand. You can definitely use your reactions if you want to. But you've also maybe heard about bacteria in food, okay? And I don't know if anybody's ever had the situation where they've eaten some bad food and then they felt like this. Bacteria also can do this. These are things called foodborne pathogens, okay? And if you've had any experience with this, it stinks, okay? And oftentimes it's either very uncomfortable or, of course, in certain situations, could be deadly. And so we are looking at one of these bacteria that's a foodborne pathogen called Bacillus cereus. Okay, this one is again an opportunistic pathogen that's transmitted generally through food and food products, plants, and other things. Okay, and it's interesting in this case because the virulence factor produced by this pathogenic bacteria is different than pneumonia. In fact, the virulence factor we see in this case is toxins. So toxins are produced by the bacteria, which is in part what makes us feel the way we do when we are infected with this bacteria. Now you'll notice the image that just popped up, you've got a lot of red, okay, on what's called a petri dish, okay, and you'll see that there's some bacteria on there that's turning white in the presence of red. That's because one of the toxins that's produced by this bacteria is something called a hemolysin. And this virulence factor actually bursts or destroys red blood cells. And so we're looking at ways in which we can stop the effects of this bacteria since we know that these virulence factors are actually controlled by quorum sensing. This, just like I didn't mention this, but our last project on pneumonia is collaborative with the University of Nevada, Reno. This project's collaborative with a professor and a graduate student at the Hebrew University in Jerusalem. I actually spent some time with them in the summer of 2019 working on this project. And the peptide that's used to regulate quorum sensing in Bacillus cereus has a name, PAP-R7. The name, of course, isn't so important. There's 7 amino acids in the peptide, but what we're really interested in and how we can change the structure of the peptide to, again, control the communication. So here's what we're doing to try to control the communication in this case. We're looking at amino acids and we're just changing one part of them. We're changing amino acids and giving them something called a methyl group. If you've had chemistry, you might recognize methyl group. If not, just know that it's like an added carbon that we're adding. So we're changing the structure again of these amino acids. And it's crazy because it looks like a really small change, even if you're just looking at the differences between the pictures, but it can have a really big effect. We made a bunch of peptides and found out that the effect was quite large in terms of the way in which we could inhibit quorum sensing in Bacillus cereus. And you don't need to look at all the numbers on these charts. I will tell you though, when the bars are small, we get excited because in this particular biological assay, when the bars are small, we're showing inhibition. And these peptides actually have IC50 values, which are just those values for inhibition again. that are pretty low. And in fact, though they're not as low as in strep pneumonia, these are the best inhibitors currently in the field for quorum sensing and Bacillus cereus. So we're really proud of that. One other thing that I'll anecdotally tell you about this project, which is kind of cool and a little bit gross, is that we also characterize, not only in a bacterial assay, but we also take these peptides in bacteria and we put them on sheep's blood. And we then kind of test to see if we're able to stop the hemolysis, those virulence factors that burst red blood cells. If the red blood cells are being killed, we see what kind of looks like a halo around this white spot. The white spot's where we put the peptide and the bacteria. As the bacteria grows, it kills the red blood cells. If we stop the bacteria from quorum sensing, even though it still may grow, it doesn't produce those virulence factors. And so we don't see any halos here on the left-hand side of the image, kind of where it says A and D. So we're actually able to use these to inhibit the virulence factors that result from quorum sensing of Bacillus cereus. And we not only see that in our initial assay, but also on this sheep blood auger, which just sounds super gross, because we can see the reduction in the halo size. So that was a really interesting thing to find as we were making these peptides. All right, so that's all for the bad bugs. Now it's your time to wake up as we close out the presentation day and respond for our last project, because it turns out it's not all doom and gloom. Okay, there are some bacteria that are not bad, and I'm curious if you know, what percentage of bacteria are harmful bacteria, ones we call pathogenic bacteria, versus those that are not harmful for us? There are some bacteria that are not harmful, so feel free to click in your answers. See, some of you guys are doing that. Awesome. Oh, this one's tough. You might be relieved in the answer. It turns out only 5% of bacteria are pathogenic. Wild, right? But they wreak a lot of havoc. Yeah, I know. Feel good about that, right? They wreak a lot of havoc. So then what's going on with the other 95% of bacteria? So real quick, a little bit of a softball question. What biological processes can good bacteria help with?
I'd say all of the above.
Pearl, you're the best. You are absolutely right. You must have been paying attention the whole time. You crushed it. And so did other people. All of the above is the correct answer. Good bacteria help us with a lot of things. And there's more bacteria cells in our body than there actually are eukaryotic cells and other cells that we would deem maybe non-bacteria or human cells. So bacteria are really important to us. So in addition to trying to inhibit the bad bacteria, we also sometimes want to encourage the good bacteria. And that's the final project we're working on here at Moravian. The two bacteria we talked about so far, strep pneumonia and Bacillus cereus, they're pathogens. We want to stay away from them. We want to try to turn them down. They're aggressive. They, especially if we get sick already, can wreak some havoc biologically on us. But then there's some bacteria like Lactobacillus plantarum and other Lactobacillus bacteria that they're good bacteria. They're cute. We like them. We call them commensal bacteria because they don't harm us. And many times these bacteria are either in our bodies already or they're in the things that we eat. If you guys have ever heard of, probiotics and things that are bacteria in yogurt, active cultures that you're eating are supposed to be good for you, that's bacteria. And in many ways, Lactobacillus plantarum is a bacteria that could really be helpful for us. I'll tell you why. Lactobacillus plantarum, it's found in foods. It is not native to the gastrointestinal tract, but because it's found in foods, we do detect it in the gastrointestinal tract of many humans. So it's kind of, in the way that it acts, considered a probiotic. And the why it's considered a probiotic is because it's immunomodulatory. Now that word's a crazy sounding word, but I bet we can piece apart what it means. What really this does is that Lactobacillus plantarum can help keep the regulation of the things we call cytokines. They're molecules that help to control our inflammatory response and our immunity response. They kind of keep them at bay. What happens is that Lactobacillus plantarum actually helps to balance out the ratios of two particular chemicals in our body called interleukin 10 and interleukin 12 to help reduce the amount of inflammation that's present in our guts. Now, why would that be bad? Well, it's because inflammation in your gut leads to a lot of negative conditions like irritable bowel syndrome and Crohn's disease and colitis. And so really what we're trying to do is increase quorum sensing, which is different from before. We do want to increase quorum sensing now in Lactobacillus plantarum. We'll see that one of the virulence factors for Lactobacillus plantarum that's controlled by quorum sensing is adherence. So the more we increase it, the more those cells can hang around in our GI tract and reduce inflammation. The more they reduce inflammation, the better we can improve our gastrointestinal health. So this is the sort of ethos of the final area of research that we're working in. And just real quickly, I'm going to show you where we are with it. This is our newest area. This is a representation of the quorum sensing circuit for this particular bacteria. I would tell you that the most important thing to pull out of this is just that the peptide is called LAMD558, okay? These little donuts that you're seeing on the screen, these are the text messages that were the triangles before, okay? And so really what we're trying to do now is encourage the production of more of these orange circles. The reason why is because the virulence factor that results is increased cell adherence and biofilm formation. And again, this keeps the bacteria in our gastrointestinal tract longer, which means that we benefit more from the effects that it has on our immune system and inflammation. So what we're doing right now in the lab is we're making some of these peptides. We're making derivatives of some of these peptides. And so you can see some of the substitutions here on the left-hand side. The peptide sequence here only has 5 amino acids, and it's actually circular, which is really interesting. It's called a cyclic peptide. We are developing an assay, a biofilm assay, which again is colored and allows us to see how much of the biofilm is produced, basically how much of the quorum sensing is turned on. The darker the purple means we have more quorum sensing. And we're actually even trying to engineer a genetically modified bacterial strain that will allow us to get either a fluorescence detection or something that again is a bright yellow color for us to measure if we are able to turn quorum sensing on or off. So with that, I think I'm going to wrap up. I think this goes to 1:15, and so we might have some time if there are any questions. I'll let Justin lead that for sure. But I do want to just say thank you to all of the students who have worked on these projects. So we also were forced into Zoom land for a little while, but we were able to get together and talk about stuff. And even on the bottom row are some of our alumni who graduated in 2018 and 2017 who surprised us on a group meeting Zoom call. We've taken students to Nevada every year to work on the strep pneumonia project with our collaborator, Dr. Talgan at the University of Nevada, Reno. Really cute tradition too is that the students in the lab, when they join and they make their first peptide, they usually tend to make baked goods to celebrate. Can't do that this year, but it's a wonderful tradition that I hope carries on post-COVID. And then here's the university in Rehovot, Israel, where I worked for a summer and we built the Bacillus cereus collaboration. So thanks to all the students who have done so much of this work and my collaborators. We have some funding sources shown here, but now I'll pause for any other questions. And thank you so much for attending. I know that this is not necessarily what anybody envisioned for the alumni engagement for this year, but I give Justin and Amanda a lot of credit for sort of making this happen. And it was an absolute pleasure for me to participate.
And thank you.
Oh, thanks, Pearl.
Thank you, Mike, so much. A pleasure having you, with us this afternoon and really showing all the incredible work that you and our current students are currently facilitating within the chemistry program. So I really appreciate you being here this afternoon. And as Mike said, if anyone has any questions for him, we do have about 15 minutes left here for this return and learn. So please feel free to put your question in the chat box. Oh, Lauren actually has a question for you, Mike. Which project are you most excited about that you and your students are working on currently?
Oh yeah, I like them all. I think that's why we keep doing them. I have to be honest though, the one because it's been the toughest to get some traction on is the one with Lactobacillus plantarum. I also think that a lot of the people who are doing research in the field have looked at the pathogens, the things that infect us as like, oh, we want to tackle these first. And that's with good reason. but I think we've got a really cool opportunity to look at the bacteria that we already have that are commensal and that are good and making them better. And I think that's just a really different way of looking at quorum sensing. So I'm pretty pumped about that one. Thanks for asking.
If you do have any additional questions, please feel free to reach out to me or Amanda at our office, and we will be able to get your questions answered for Mike. Thank you all for being a part of this presentation, and we hope to see you at our STARS student panel at 2 o'clock.